Glycolysis is the metabolic process that serves as the foundation for both aerobic and anaerobic cellular respiration. In glycolysis, glucose is converted into pyruvate. Glucose is a six-membered ring molecule found in the blood and is usually a result of the breakdown of carbohydrates into sugars. It enters cells through specific transport proteins that move it from outside the cell into the cell's cytosol. All of the glycolytic enzymes are found in the cytosol.
The first step in glycolysis is the conversion of D-glucose into glucose 6-phosphate. The enzyme that catalyzes this reaction is hexokinase. Here, the glucose ring is phosphorylated. Phosphorylation is the process of adding a phosphate group to a molecule derived from ATP. As a result, at this point in glycolysis, one molecule of ATP has been consumed. The reaction occurs with the help of the enzyme hexokinase, an enzyme that catalyzes the phosphorylation of many six-membered glucose-like ring structures. Atomic magnesium is also involved to help shield the negative charges from the phosphate groups on the ATP molecule. The result of this phosphorylation is a molecule called glucose 6-phosphate, thusly called because the 6th carbon of the glucose requires the phosphate group. The second step of glycolysis involves the conversion of glucose 6-phosphate to fructose 6-phosphate. This reaction occurs with the help of the enzyme phosphoglucose isomerase. As the name of the enzyme suggests, this reaction involves an isomerization reaction. The reaction involves the rearrangement of the carbon-oxygen bond to transform the six-membered ring into a five-membered ring. The rearrangement takes place when the six-membered ring opens and then closes in such a way that the first carbon becomes now external to the ring. Step 3. Phosphofructokinase, with magnesium as a cofactor, changes fructose 6-phosphate into fructose 1,6-bisphosphate. In the third step of glycolysis, fructose 6-phosphate is converted to fructose 1,6-bisphosphate. Similar to the reaction that occurs in step 1 of glycolysis, a second molecule of ATP provides the phosphate group that is added onto the fructose 6-phosphate molecule. The enzyme that catalyzes this reaction is phosphofructokinase. As in step 1, a magnesium atom is involved to help shield negative charges. The fourth step utilizes the enzyme aldolase, which catalyzes the cleavage of fructose bisphosphate to yield two 3-carbon molecules. One of these molecules is called glyceraldehyde 3-phosphate, and the other is called dihydroxyacetone phosphate. In step 5, the glyceraldehyde phosphate is the only molecule that continues in the glycolytic pathway. As a result, all of the dihydroxyacetone phosphate molecules produced are further acted on by the enzyme phosphate isomerase, which reorganizes the dihydroxyacetone phosphate into glyceraldehyde phosphate, so it can continue in the glycolysis. At this point in the glycolytic pathway, we have two 3-carbon molecules, but have not yet fully converted glucose into pyruvate. Now in this step, two main events takes place. First event, glyceraldehyde 3-phosphate is oxidized by the coenzyme nicotinamide adenine dinucleotide. The second event, the molecule is phosphorylated by the addition of a free phosphate group. The enzyme that catalyzes this reaction is glyceraldehyde 3-phosphate dehydrogenase. The enzyme glyceraldehyde phosphate dehydrogenase contains appropriate structures and holds the molecule in a conformation such that it allows the NAD molecule to pull a hydrogen of the glyceraldehyde phosphate, converting the NAD to NADH. The phosphate group then attacks the glyceraldehyde phosphate molecule and releases it from the enzyme to yield 1,3-bisphoglycerate, NADH, and a hydrogen atom. Now in this step, 1,3-bisphoglycerate is converted to 3-phosphoglycerate by the enzyme phosphoglycerate kinase. This reaction involves the loss of a phosphate group from the starting material. The phosphate group is transferred to a molecule of ATP that yields our first molecule of ATP. Since we actually have two molecules of 1,3-bisphoglycerate because there were two 3-carbon products from stage 1 of glycolysis, we actually synthesize two molecules of ATP at this step. With this synthesis of ATP, we have cancelled the first two molecules of ATP that we used, leaving us with a net of 0 ATP molecules up to this stage of glycolysis. Again, we see that an atom of magnesium is involved to shield the negative charges on the phosphate groups of the ATP molecule. Step 8 involves a simple rearrangement of the position of the phosphate group on the 3-phosphoglycerate molecule, making it 2-phosphoglycerate. The molecule responsible for catalyzing this reaction is called phosphoglycerate mutase. A mutase is an enzyme that catalyzes the transfer of a functional group from one position on a molecule to another. The reaction mechanism proceeds by first adding an additional phosphate group to the 2 position of the 3-phosphoglycerate. The enzyme then removes the phosphate from the 3 position, leaving just the 2 position phosphate, and thus yielding 2-phosphoglycerate. In this way, the enzyme is also restored to its original phosphorylated state. This step involves the conversion of 2-phosphoglycerate to phosphophenol pyruvate. The reaction is catalyzed by the enzyme enolase. Enolase works by removing a water group or dehydrating the 2-phosphoglycerate. The final step of glycolysis converts phosphophenol pyruvate into pyruvate with the help of the enzyme pyruvate kinase. As the enzyme's name suggests, this reaction involves the transfer of a phosphate group. The phosphate group attached to the 2-carbon of the phosphophenol pyruvate is transferred to a molecule of ADP, yielding ATP. Again, since there are 2 molecules of phosphophenol pyruvate, here we actually generate 2 ATP molecules.